So I'd like to begin by telling you a little bit about myself and my background, because after all, scientists are people. My story starts in Panama, actually, which is where my father was born, in the littlest guy in this black and white photo here. He was born in Panama in 1959 and immigrated to the United States to Brooklyn and grew up in Brooklyn and in Maryland, where he met my mom, who's pictured in the photo below. And I was raised in Maryland, in Silver Spring, a suburb of Washington, D.C. And this is my family in the picture on the bottom right. I have my two parents and my younger brother. So I attended a high school in Silver Spring, Maryland called John F. Kennedy High School. And there is where I started to meet some of the people that really influenced my trajectory and my decision to become a scientist. So I had an AP English teacher named Mr. Howard that really helped to foster my love for learning. An AP Calculus teacher named Miss Davis, who really encouraged me to be ambitious in my pursuit of knowledge and where I might end up going to college. And finally, my AP Biology teacher named Miss Cush. And Miss Cush is actually the person that led to me meeting Professor Walker, who you will meet in a little while, because she encouraged me to attend a course just like this one at the Howard Hughes Medical Institute. And this one was called AIDS: Evolution of an Epidemic. And one of the speakers of that course was, of course, Dr. Walker, who you will hear a lot from in the coming lectures. And he spoke about people called HIV elite controllers, who are people who get infected with the HIV virus but never progress to AIDS. In other words, their immune system spontaneously controls the virus. And as an immunologist, Dr. Walker was hoping to study these folks to try and understand how their immune system fights HIV in order to help develop vaccines for the rest of us in order to protect the entire globe from HIV. And so this idea that a basic scientist could use the scientific method to potentially save millions of lives really struck a chord with me. And before this, I had no idea that science was a career. But after seeing this lecture by Dr. Walker, I decided, okay, I'd like to become a scientist and study the immune system when I grow up. But of course, that was still a long path. And so there were a lot of other people that helped me to get where I am today. So after seeing this lecture from Dr. Walker and deciding that I wanted to become a scientist, I was able to get an internship in the laboratory of Dr. Susan Ray, who worked at the National Institutes of Health, which is also located in Maryland. And so this is a picture of me working in my first laboratory, and I had such a powerful experience there and just found being in the laboratory so fun, and so decided this is something that I wanted to continue when I was an undergraduate student. And so in addition to wanting to try to help people through science, I also was really taken with the idea of global health and all the different ways in which we can try to improve the health of people around the world. And so in particular, I was excited by the idea of trying to improve things for folks that were living with HIV. And so in addition to my research, I got involved with a group called the Harvard Global Health and AIDS Coalition, which tried to advocate for increased access to medicines in the developing world, as well as for increased funding for HIV, tuberculosis, and malaria, these really terrible pathogens that affect millions of people around the world. But in addition to this, I also continued to do research. And so here I was again lucky to meet lots of mentors who supported me. I was first invited by Professor Richard Losick, a molecular and cellular biology professor, to join a mentoring program for undergraduates called HHMI Ideas. And this program helped pay me in order to be able to work in the laboratory and do my research so that I didn't have to have a job on campus. And so the laboratory I worked in was run by Dr. Eric Rubin at the School of Public Health, where I was mentored by a then graduate student, Mandy Martineau, who both of them were working to try and understand how tuberculosis causes disease. And so through this experience, I got to work on my first project in a lab and really confirmed doing research is something that I want to do for my career. And while I was in college, I also had the opportunity to travel to do research. And so I spent a summer living in Portugal studying malaria, another one of these really important global pathogens. And this really excited me because I learned that I could do this research that could potentially help people. I could do science, which I absolutely loved. And I could also live around the world and meet people from all different countries while doing all of these things. And so that was what really allowed me to then take the leap after graduating from college to go and live in South Africa for 2 years, where I worked at the Africa Health Research Institute, which was then called KRIF, with Dr. Emily Wong and Dr. Thumbi Ndungu, where I did research on HIV and tuberculosis infection in humans. And this is what really solidified my excitement for the immune system and understanding how the immune system helps protect the body from infection. And so after leaving there, I went on to get a PhD in immunology at Yale University, where I was mentored by Dr. Ruslan Medzatov. And at Yale, in addition to studying the immune system at a more basic level, I was also really interested in helping to shape the face of science to become more diverse, helping to communicate science with the rest of the general public, and also helping to get younger people interested in science through outreach. And so I was involved in all these various different initiatives, like writing articles published online, holding a science fair for middle schoolers, starting a diversity initiative in my department, all of these things that I'm really passionate about, in addition to my scientific research. And so that's just a bit of the background to how I got to where I am today. And I'm now a postdoctoral researcher at Harvard. So I've gotten my PhD, and now I'm continuing to do research on the immune system, and specifically about how the immune system affects the brain. Now we're going to start the first lecture, which is recognizing a new disease. So I want you to think back to December 2019, which is when COVID-19 emerged as a new disease. Back then, what appeared was a pneumonia of unknown cause. And so in this X-ray image, what you can see here is on the left a depiction of a normal lung, and on the right a depiction of a lung with pneumonia. And so you can see all this cloudiness that makes it difficult for people to breathe. And so this pneumonia of unknown cause emerged in Wuhan, China in December of 2019. Over the course of the next month or so, the cases of severe pneumonia of unknown cause in Wuhan, China just skyrocketed. And while some of them were linked to this Huanan market, and eventually there were the emergence of cases that were not linked to this market, indicating that this pneumonia disease was spreading throughout the community. And this suggested that, of course, it might be caused by an infectious pathogen. And this first public report of this new disease occurred on December 27th, 2019. And in less than a month, scientists were able to identify the virus called SARS-CoV-2 as the causative agents of this new pneumonia disease. Less than a month later, scientists were able to identify SARS-CoV-2 virus as the causative agent of this new pneumonia disease. So in January 2020, scientists, as I mentioned, identified the SARS-CoV-2 virus. And this has a few important components that I want to point out to you that'll become important later. First, there's the RNA inside this capsid, and this is the genetic code for the virus in the same way that DNA is our genetic code. And then surrounding this capsid is the spike protein, which is how the virus enters our cells. And we'll get into this a lot more in the second lecture. Now, SARS-CoV-2 is not the first deadly human coronavirus. And there have been a few coronaviruses throughout the past several decades that have infected the human population. So there was first the SARS virus in 2003 that infected on the order of 8,000 people and had a much higher lethality than SARS-CoV-2, about 11%, but a much lower transmissibility. Then there was the MERS virus in 2011, which resulted in about 2,500 cases and had a much higher lethality rate of 34%, but again was much less transmissible. And what we're experiencing now is the pandemic caused by SARS-CoV-2 that emerged in 2019, which has infected a huge amount of people. It has a much greater transmissibility than these other coronaviruses, but has a lower lethality. Something we want you to continue thinking about throughout this course is the potential for new coronaviruses to emerge that have the same high transmissibility as SARS-CoV-2, but an even higher lethality akin to MERS or SARS. And so it's really important for us to be able to identify and strategize about how to combat future pandemics in order to prevent something like this from emerging and causing a huge catastrophe for the human population. Now, as you all know, COVID-19 has spread rapidly across the globe. And there are a few things I want to point out in this map that might be important to consider. So first of all, the number of global cases is absolutely enormous, and there's been a huge and tragic loss of life. There's something else that I'd like you to notice about this map, which is that there are certain parts of the world that are less affected by COVID-19, including Australia, New Zealand, and other parts of Asia. And that's in comparison to the United States and Western Europe, where the COVID-19 pandemic has really exploded. And so I want you to be thinking about the differences between how different countries have approached the control of the pandemic and how that then results in this huge difference in cases and deaths across the globe. And in addition to differences between countries, we of course know that there are also huge differences in how COVID-19 has affected different populations in the United States. And so the hospitalization rates for Latinx, Indigenous, and Black folks that are infected with COVID-19 are much greater than those for the white population. And the same goes for the rate of death amongst these communities of color. There are a number of factors that contribute to these disparities, including access to healthcare, the prevalence of comorbidities, and so these social determinants of health are things that we'd like you to continue thinking about when thinking about all the ways in which a pandemic disease can move through a population. So to take a step back and talk more about SARS-CoV-2 itself, we want to walk you through what actually is a virus. And so viruses and bacteria are pathogens. These are things that can cause disease in our body. So one example of a bacterium that you might be familiar with is E. coli. This can cause things like food poisoning. And a big difference between bacteria and viruses is their size. And so this scale really shows you that these viruses are much, much smaller than bacterial cells, and even there's an even greater difference between the size of a virus and the size of our cells. So what's really remarkable about viruses is that despite having only 10 genes, they're able to co-opt the entire physiology of a human, which has 20,000 genes, and cause the pandemic that we've seen occur over the past year and a half. Stepping back again, we want to define what a pandemic is. And this is an infectious disease that is spread over several countries or continents that usually affects a large number of people. And that's, of course, exactly what we're dealing with COVID-19. And some historical examples of pandemics include the Black Death, the Spanish flu, the HIV pandemic, and the smallpox pandemic. So where did SARS-CoV-2 actually come from? Now, this is a question that scientists are still actively investigating, but one theory is that SARS-CoV-2 came from animals. And so there's some evidence that there was transmission of coronaviruses between horseshoe bats and pangolins in China, and that these viruses were evolving between these two animals and then eventually jumped into the human population. And so scientists are working to understand whether the virus came from animals or whether it might have come from another source. And so this is an example of a zoonosis, which means when viruses go from an animal into the human population. And there are actually a lot of other examples of zoonotic viruses. So HIV is probably the most famous one, which came from chimpanzees. And there's also cases of influenza that have come from animals. There's the swine flu, as well as avian flu, and that came from pigs and from chickens, respectively. It's really important to be thinking about surveillance of these animal reservoirs of emerging viruses in order to combat and identify future pandemics from other coronaviruses and other zoonotic infections. So it's useful to compare these two most recent pandemics of SARS-CoV-2 and HIV. Now, SARS-CoV-2 has caused over 140 million infections in the course of 1 year. And HIV, in contrast, has caused 76 million infections in 40 years. So how did SARS-CoV-2 infect so many people so quickly? One of the biggest differences between SARS-CoV-2 and other viruses is that it infects people when they're asymptomatic. So when we look at things like influenza and SARS-CoV-1, the peak of transmission occurs when folks are symptomatic. So this means you're already feeling sick, and that's when you're probably going to transmit the virus. And so hopefully you're staying home, you're resting up, and you're not transmitting it to other people that you're interacting with. In contrast, SARS-CoV-2 is the most transmissible when people are asymptomatic. And so this means that before you start to feel sick, while you're out and interacting with people in the world, you're transmitting virus to them. And so we think that this is one of the major reasons why SARS-CoV-2 spreads so rapidly throughout the population. So SARS-CoV-2 is transmitted through the air, through the respiratory route. It can travel in droplets, which are 5 to 10 microns in size. And it can also travel in aerosols, which are much smaller particles, less than 5 microns, and these can stay in the air for hours after a person has left the room. And so this is another reason why SARS-CoV-2 has been able to become so transmissible. And so given that we have this high rate of transmissibility when people are asymptomatic, it's important to be able to rapidly diagnose people with SARS-CoV-2 infection. And so there are a number of different tests for SARS-CoV-2. One of them is the test for the virus genetic material. And this is a PCR, or polymerase chain reaction based test. So this detects the SARS-CoV-2 viral RNA. So what does a positive PCR mean? And what are the CDC guidelines for quarantining when you've tested positive? So the graph here shows the percentage of people who will test positive versus the days of their symptom onset. And then this little triangle that's inset here shows how infectious a person is. What you can see is that this PCR test can detect people when they're the most infectious, but before their symptoms have begun. And so this is a really important tool for pandemic control. Another question, though, is whether any test can differentiate whether someone is infectious and whether they're not. And so there's another type of test called antigen tests that detect viral proteins, such as the spike protein that's on the virus surface. And so these are less accurate than PCR tests, but they're a really useful tool because they're more rapid and can help to control the pandemic and help people inform their behavior if they have a positive test. These rapid and affordable COVID-19 diagnostic tests, such as the antigen-based tests, are a potentially really useful tool for us to be able to use to curtail this current pandemic and future pandemics, because this can function more like an at-home pregnancy test. It's the type of thing that you could do before you're going to leave the house for the day and find out if you're infected, and then you can adjust your behavior accordingly. And so tools like this and technology development is a really useful strategy for combating future pandemics. So how do we stop a pandemic? Number one, we prevent the spread. Number two, we treat infected patients, which we'll talk about in the third lecture. And number three, we can develop vaccines, which we'll talk about in the fourth lecture. And how do we prevent the next pandemic? We use surveillance networks to detect whether animals are harboring these zoonotic viruses and whether new viruses are entering the human population. We can contain the initial virus as it emerges through rapid detection and containment. And then finally, we can develop these prophylactic vaccines in order to protect people from subsequent infection. So a question that you might be wondering about is, can we ever eradicate SARS-CoV-2? It went from bats into pangolins into humans, and now we have, luckily, the proliferation of vaccines in the United States, and they're beginning to spread around the world. So will we ever be able to fully eradicate this virus? And the answer is, unfortunately, probably not, because there's now evidence that the virus can move from humans into other animals, such as minks, which are depicted here, and then back into humans. And so this means that the virus can, again, stick around in animal populations and then return into the human population, which means that we'll probably not be able to ever fully eliminate SARS-CoV-2. But we can, of course, try and control its spread and mitigate the damage that it causes to the population. So to summarize this first lecture about identifying a new disease, I've shown you that viruses can be an existential threat to humanity. Again, thinking about a virus that's as lethal as MERS, but as transmissible as SARS-CoV-2, this could be, of course, an existential threat to the human species. So it's important to think about how to control future pandemics and to understand viruses more. Cross-species transmission of virus is increasing. As humans encroach on wild animal populations, these viruses that tend to live in animal reservoirs are coming into more contact with humans, and so these zoonoses are increasing in frequency. Pre-symptomatic transmission of a virus accelerates spread. So one of the major reasons why SARS-CoV-2 was able to spread so rapidly throughout the world is that people are more transmissible when they don't have symptoms. And so the proliferation of rapid diagnostic tests is critical for controlling a pandemic such as this. Thank you so much for watching, and we hope to see you in the future lectures.